The title really, I mean, when you look at what I have in there, you probably think of biodiesel, soybeans. And today I'm going to talk about biodiesel processes just as an example of the many things that we do in our laboratory to use microbes and harness their activities for applications, practical applications. And the two pieces in the pod that I'm going to talk about first are actually the two major issues that we address, water and energy. But you'll see the two pieces going on all the way till the end. Why are we interested in water and energy? They are interlinked. They are so intimately connected that you cannot have one without the other. And this is what I try to show here. We need water for energy. At all steps, we need it. Think about a nuclear plant. The amount of water that is used for cooling, for example, something as simple as that. And then we need energy to extract water, but also to reuse it, so we can keep recycling this limited amount of resource that we have in the planet. So the problem with this strategy is that it's not always a closed loop. There is a lot of waste generated along the way, and there is a lot of diverted energy to get water and water for energy that could be improved. This is just from the World Bank, an estimate to give you an idea of something that we all have in our minds, but seeing it in numbers helps us consider the problem here. By 2035, energy consumption in the world is expected to increase 35% already of the consumption right now. And this is going to come with an 85% increase in water consumption. So it's not even proportional. We're always going to need far more water to generate energy and satisfy our energy needs than we need the energy itself. So there is a lot of pressure on water reuse, finding ways in which we can reuse the water as many times as we get the energy, but also in reducing the amount of energy that we need to use to regenerate that water. So going back to this cycle that I just showed you, this is the part in which I want to stop for a while, and it's all the waste in the form of wastewater that is going to be generated at all steps in this cycle that we are interested in, and its treatment. The reason why is because when you consider the amount of waste that we generate during energy production, there is always going to be a substantial amount of energy that is actually used to treat that waste. Think about the example of a biodiesel plant, since I'm going to talk quite a bit about it today. We need water to make biodiesel, a lot of water to make it. And one of the problems with biodiesel right now is that the wastewater that is produced has actually a lot of organic matter still holding a lot of energy that doesn't go into biodiesel. That's glycerol. We call this usually glycerin wastewater. The practice for many years was to concentrate that waste, to remove the water, and purify the glycerol at least to 85% so it could be sold to glycerol refineries. The problem is that as we're moving into a bioeconomy, producing more biofuels, we have a glycerol glut. There is so much glycerol being produced in biodiesel. Think about bioethanol refineries that produce 10% of the plant biomass that is fermented to make bioethanol. Actually comes as glycerol that is also a waste. So there is simply the prices of glycerol have dropped so much that many biodiesel plants don't have profits if they follow this route. But then they have a big problem because the glycerin wastewater is a hazardous material. It still has traces of methanol that they use in the first step. They actually have to pay a high fee to wastewater treatment plants to clean it up. So not only they are not getting the profit that they were getting before, they actually have to pay a lot of money to get rid of that wastewater. So it has become an economic and also an environmental liability, because if they dump it in the environment, many problems are also caused because of the hazardous waste. So this is just an example. So what do you have to do? You have to get a lot of energy that comes from the potential profits of the company to treat this type of wastewater. And there are many different alternatives, but none have really provided the profits that are necessary for the biodiesel plants to survive. And this is the reason why you see the examples here in Michigan. Many small to medium scale biodiesel plants have closed. Even with subsidies, they simply cannot survive. So this is one of the problems that we like to address. To be able to have a sustainable process in which we can add a second step, in which we can actually harvest the energy in the glycerol in here, and basically clean up the wastewater, remove every single piece of organic material that is contaminating it, get the energy, and at the same time reduce the water, clean it up. So what we are doing is actually generating energy that goes back into the system rather than the reverse. And where did we look for alternatives? We went to the cow. We actually are environmental microbiologists, and we love to go to the environment and see where in nature these processes that we are going to try to introduce in an industrial setup, which ones are performed by microbes naturally. And so it happens that inside the rumen of the cow are many of these very interesting processes that we like to harvest. Here in the rumen is where all the plant biomass is going to be processed, and where many of these waste products of industrial plants are actually going to be further metabolized to extract all the energy. So the cow doesn't have any waste in the end. It uses it all. And this is actually what we are looking up. Here, I don't know if you are familiar with this, but there are actually systems in many universities where you actually do surgery on the cows, and you have, I don't know if you've seen the glove bags. Bill was showing me some of the laboratories that you have here, the glove enclosures that have these big gloves, so they're always prevented from exposures. This is exactly what you have. It's like going into a glove bag, but you actually go into the rumen of the cow, and you can get samples directly from them. And the cows don't suffer. They complain a little bit when you go in and out, but you get actual samples of all the microbes that are really in this natural reactor that is the cow rumen. And when you look at them even under the microscope, you get a sense of just how much diversity of microbes are there working together to make sure that no matter how much different plants are being digested, everything gets digested by the cow and everything, all the energy is harvested. So this is actually a very, very simplified scheme of what's going on in the cow. If you look at textbooks, you have a heart attack because of the level of detail, the many activities that are here. But the important ones is that there is a diverse substrate intake in plant biomass. And these plants, these cow rumen microbes are going to hydrolyze those solids, and they are going to generate sugars that they can ferment. This is exactly what our saliva does when we're chewing a candy. A solid, and we're extracting the soluble sugar so we can metabolize them. That's basically it. So you are probably most familiar with this. This is the way ethanol is made, for example, in a bioethanol refinery. But there is also a lot of byproducts. And there is nothing we can do about these byproducts. Microbes need to make byproducts because that's how they generate energy for growth. So every time you have a biofuel of interest, you are going to also produce these byproducts. But in the cow rumen, you have a lot of many different microbes that specialize in that waste. And they metabolize it, and they make something useful. In the cow rumen, what the cow wants is propionate because it feeds its muscles with it. It's the only thing that the cow cannot make, and these microbes make it for it. Very much like our vitamins. The ones that we cannot make ourselves, we have vitamin pills, and that's our. These guys have microbes. And then they make methane. And I don't need to tell you where the methane comes out. You already know what it is. But something that is very interesting is that glycerol is also part of the diet. Not only that, some of the first microbes that ferment the sugars make glycerol, pretty much like yeast in bioethanol refineries also make glycerol as a byproduct. So the cow rumen is a reactor that has naturally evolved metabolisms to digest the glycerol. So what we wonder is whether we can actually look at this very diverse microbial population and find microbes that we can use to treat glycerin wastewater, for example. What most people don't know, and this is where my lab expertise comes, is that every time you have a process of dissimilation like this one, electrons are being extracted from the organic material. So there are electric currents that we don't detect, but the microbes are really extracting electrons and passing it on to other things. It could be oxygen, they also respire like us, or it could be any other compound other than oxygen. So these electron currents could potentially be harvested as electricity as well, and this is what we do in my lab at the same time. So if instead of the cow having muscles to feed on this propionate, we had an electrode, I can guarantee that we will measure an electrical current, because at the very bottom we will have organisms that are able to extract electrons from the hydrogen and dump the protons, and there will be some that are able to get each of the different organic acids and separate the CO2 and get the electrons and dump them onto something, in this case an electron, and so on. So if we focus on the reactions of interest, what my lab decided was to look at this as a two-step process. Can we identify an organism, the one that processes the glycerol, and from now on I'm going to call it a glycerol microbe, or GM, and then can we find organisms that are specialized at selectively removing these waste products of the fermentation and harvest all the energy to make electricity. So this is the two steps. And this is the microbe that I'm going to call the electrogenic or electrogen microbe. And in some cases I abbreviate it as electro. That sounds like a comic. So here is the electrogenic microbe that my lab has many projects with. This is called Geobacter. It's a bacterium from Earth. That's what the Latin word means. You can find it almost everywhere because they respire not oxygen. They actually don't like it very much. They respire iron oxides, minerals, rust. That's what they like to do. And as you know, if you've ever seen rust on a metal nail, rust is actually a solid, very much like an electrode is. And what these organisms have done is, this is supposed to be a very ancient process that probably originated in Earth billions of years ago. So they've had a lot of time to perfect the process. What they have marveled is at transferring electrons from all the metabolism, the digestion of their food, to the electrons, transfer the electrons exactly outside to these minerals. And when they do this, they actually solubilize some of the iron, but they also make a mineral that I'm sure all of you are familiar with, magnetite. And there is this idea that the magnetite they make is very unique. And there are meteorites, like one that they examined that came from Mars in the 80s, that have actually magnetite that has exactly the same signatures as the magnetite that these microbes make. So this opens many possibilities also of whether these are processes that are really more widespread, even outside our planet. But the important thing to us is that magnetite is a semiconductive material. And what we know is that even after they dump electrons to the iron minerals and they make magnetite, they remain attached, and they use it to dump electrons to another microbe that is able to get it from the magnetite. So it's as if you have an electrode with one microbe on one side making the electrons and another receiving them. And that's what happens in the environment with all these minerals. So what we decided to do is, okay, how promiscuous are they? Can we give them an electrode with very similar electron accepting capabilities as the natural minerals, and can they make electricity in a fuel cell? And the answer is yes. This is an example of some of these surface-attached communities that we call biofilms that are generating electricity because they're dumping electrons onto the electrode. And I don't know if you can see, but there are a lot of fibers. My lab has been studying how these organisms have formed these power networks, like a nanogrid. They actually make protein filaments that are conductive, nanowires, which we are exploiting for many applications. Today, unfortunately, I'm not going to tell you about it. I didn't realize that I had a fellow electrochemist like Tom here that I could chat. That's for the next one, maybe. But the idea is that we have an organism that we can harvest here on electrodes, and that uses exactly the fermentation byproducts that we're trying to get rid of. And the one we are using doesn't like ethanol. So it's perfect if you want to clean up in ethanol fermentation, because it will use up everything else and leave the ethanol in the water. Just that. So I'll tell you a little bit more about it. What we've done also is find ways in which we can stack microbes on the electrode. Because they have these wires, we found ways in which we can make many different layers. And the more layers, the more current that we are producing. The more fermentation substrates that we are metabolizing as well. To the point that we are making electrodes that we hope eventually to manufacture and commercialize and sell them in dry form. So anybody could get one of these electrodes with our bugs, put them in water and just have this type of cleaning action in any type of fermentation reaction, for example. So as a proof of concept, we developed a partnership. Remember, there are two peas in the pot. We work a lot with one of them, but why not have a microbe that fits our electrogen? And in this particular example, we worked by trying to think if we could couple corn stover, which is a waste, an agricultural waste. Can we couple it to all the way to electricity? And to prove that we can make a biofuel, can we just make ethanol as the only product in this system? And we chose corn stover because we met Bruce Dale, which I'm sure most of you may know. I think he was actually a speaker here. Bruce Dale is a chemical engineer that has developed a great chemical pretreatment of this very recalcitrant substrate. So it's more digestible, and you can use it in biorefineries for fermentations. In biorefineries, they just add a cocktail of enzymes to degrade it. But what we did is use this corn stover to actually screen all our culture collection and see if we could find the microbe that was able to digest this corn stover so efficiently and ferment all the sugars and produce exactly what we want to feed our electrogen. And we found several. One of them in particular was the most suitable. I'm going to refer to it as the corn stover microbe here. But when you put them together, it was like a perfect tango. That was magic. The moment they started to grow together, there was never an accumulation of byproducts in the medium. We could barely detect them at any given time because each one was feeding each other and helping each other grow as much as possible. If these byproducts accumulate, the pH drops. They have a feedback inhibition of the activities of these microbes. Everything can collapse. But if they are producing and feeding these microbes, things actually happen even better. And what we did with this partnership of microbes was really couple the corn stover, what we call the consolidated bioprocessing of corn stover all the way to electricity and ethanol. And this is just data so you get an idea of how good it is to work together. If we use just this corn stover microbe, this is more or less how much energy we are recovering from the original substrate, the corn stover, in the fermentation. Just the two graphs are just because when you put it in a regular reactor or when you put it in an electrochemical reactor with an electrode. But there is no electrogen, so nothing else is happening. So everything stops here. And this is the amount of energy they can process, about 30%. When you put an electrode with our electrogen to soak up all these byproducts, look at the amount of energy that we are generating only in a single reactor with fermentation. And if you count the amount of electricity that you are also generating at the same time, you increase the energy recoveries even further up to above 70%. And this is without optimizing these organisms, because we are microbial geneticists. We like to do engineering, optimize the process. This is the natural process. All we did was put the two together, but we tailored a combination to make sure that each organism was helping the other one perfectly. So this was our proof of concept. Ethanol was the only thing left out in the fermentation broth, which is very important when you're trying to distill a desired product. You don't have to have a very complex separation procedure as if you have byproducts in the medium, because all you have here is ethanol and the water. So we decided to say, okay, in theory we can really customize any process we want, having this type of electrodes in a reactor fed by our electrogenic microbe. So any fermentation in theory could benefit from this process. And this is exactly what we're doing. We have a commercialization grant. It's the MTRAC program that is from MSU and the Michigan Economic Development Corporation to scale up reactors. We are trying to retrofit the standard fermenters. That's why I was so delighted when Bill gave me a tour to see really what scaling up means, as you guys are doing here in the Institute. And what we are doing is really trying to get as many different fermenters as are commercially available. And we're trying to retrofit them in a way in which we can have the electrochemical processes with the electrodes with the normal fermentation that takes place in this type of vessels. And this is just an example of a typical fermenter that we use. We have our electrogenic microbe growing on an electrode, and we actually wrap around the electrode so nothing interferes with the volume of the fermentation. And in this particular electrochemical reactors, we have a second electrode, what we call the cathode. So rather than producing electricity, we are bringing all the electrons here, and they are getting all the protons that keep being produced in the medium to make hydrogen. So this is what we call an electrolysis cell, a microbial electrolysis cell. So we are making a biofuel or an added value product here, and all these byproducts, all this waste, is being converted into CO2 and then hydrogen as well. So let's go back to the glycerol. Let me tell you how we could apply this very same proof of concept to get a reactor in which we could digest the glycerol and make an added value product. What we did is exactly the same thing as we did in the previous example. In this case, the substrate was glycerol. So can we find in this cow rumen the microbe that gets that glycerol, that metabolizes it by fermentation? So all the fermentations are going to make this type of compounds. Can we find the right one that has the right compounds to feed our microbe? And if not, can we use genetic engineering, which we love to do, to actually modify one or the other to really make the perfect matchmaking? And this is what we did. Again, this is what happens in biodiesel. You have the oil in the catalysis with salt, and with methanol or ethanol, you make the biodiesel and you generate the glycerol. So if we can put this in a fermenter to make the ethanol, we can actually recycle it in the process, creating the feedstock that we use in the first step. The only reason why U.S. biodiesel plants use methanol is because it's cheaper, but it's made from petroleum. So it is not sustainable and it's not independent of petroleum. Brazil, that makes so much ethanol, actually uses it for biodiesel, and is the only biodiesel system that is independent of petroleum, for example. So what we were proposing was to have this type of cyclic system, but also reusing the water. The idea is that ethanol is the only thing that is left in the medium, so it's very easy to distill the ethanol. And we have a water of quality that we can reduce in the process. So what I'm going to tell you is the work of two graduate students. I always bring it up because people think that we have an army of postdocs or highly trained scientists. These were two graduate students that started in the first year with me, and each one decided to devote most of their attention to one of the microbes. I told you about the two peas in the pod. These were the best two peas that I had in my lab for the longest time. So Gemma decided to work on identifying the glycerol microbe and optimize it genetically and at the genome level to make it the best possible microbe for our process. And Allison, another graduate student, took the task of getting this whole setup with the electrogen to do the matchmaking. And together they did something wonderful. This is just to see how teamwork, both for the graduate student and for the microbes. When they put the two together and they compared with just the glycerol microbe alone, you can see the fermentation products. We have a good microbe that makes quite a bit of ethanol from glycerol, but it also makes all these fermentation byproducts. We put it together with the electrogen, and the electrogen uses almost everything except lactate. So this is the first thing they say, okay, we have to manipulate either one or the two so we can get that lactate out of the way. And we did it. I'll show you the data in a little bit. But the important thing is that we were very close to having that perfect magic between the two together. So this was very encouraging for us. And we were getting, this is in millimolar, but just to give you an idea, we were getting very, very high conversion rates of glycerol into ethanol, very close to the maximum theoretical possible. As I mentioned to you, some of these byproducts are required because the cells need energy for growth. But the rest was mostly converted into ethanol naturally. So it was a good organism to have. But this is how we improved, for example, the glycerol microbe. The first thing is that we could only feed very low concentrations of glycerol because in the cow rumen, glycerol is competitively taken by many microbes, so they are never exposed to higher concentrations. So we had to make it like it better. And this is just an example of how it started to die on us as we increased ethanol, the natural microbe, and how we adaptively evolved it to love it. And it ended up loving it so much that I was like an addict. We couldn't feed this microbe anything else. No glucose, no other sugar. All of a sudden we made a very specialized metabolic route for glycerol only. But that's what we wanted, so we didn't care much about the rest. And it actually ended up getting high tolerance to many other things that are usually important to consider in scaled-up fermentations. It had high tolerance to acidic pH, and naturally it stopped producing lactate, which was wonderful because it was really our problem that our electrogenic microbe could not use lactate very efficiently. So it was perfect. We made a superbug. Alison, my other student, actually worked on improving the electrogen. And this one, what we did is actually evolve it for tolerance to ethanol. Because glycerol is a sugar alcohol, and we noticed that it was the alcohol part that was killing our electrogen. So she adaptively evolved this organism to increase its natural tolerance to ethanol. And in doing so, she actually increased the tolerance to glycerol as well. And this is what you see in this graph, that actually continues to grow. Here it dies all the way at these concentrations of glycerol, but it continues to grow. Something else that I'm not going to show you the data for is that once we grow it as this biofilm on electrode, its resistance goes off the roof, because it has all these layers, like a cement surrounding the cells, so it's even more resistant to glycerol. So we had two superb strains ready to work together. So when we put them together, this is what happened. As we increased the amount of glycerol we fed them, we increased glycerol consumption proportionally. And proportionally, we increased the amount of ethanol in a very close 1-to-1 ratio. Usually 90% of the glycerol was converted into ethanol, maximizing really the yields. But what I want to tell you, although these results were great and very promising, look at the limitations. We started very well, but we could have done much, much better. And this is, in industrial processes, this is what you want, to push the process to the maximum yields possible. And we started to identify some things that definitely need improvement. This is an example of what happens. This is the glycerol microbe. Look how poorly it performs with glycerol alone. This is the amount of ethanol in green. And what we found is that when we did our electrochemical reactor with the two together, we of course increased a lot of ethanol, but a lot of things were in here that were not ethanol, and that could potentially be ethanol. So there were limitations. This is the amount of current. And to us, this was a red flag that our electrogen microbe was not doing very well, because it was not producing as much current as it was theoretically possible, and it was not using everything else that was produced. Basically, it was indigested. We literally we increased the capacity of glycerol fermentation so much without changing the amount of electrogens, which was limited to a small electrode, that we simply overwhelmed. It was food poison. That's exactly what we did to the poor electrogen. And as electrochemists know, the surface of the electrode matters for many applications. But as you scale up a process, you have to scale up the electrodes proportionally. And we didn't do this. This was a constant in our system. We didn't want to add more variables. So what we are doing now is actually making very, very large for us, not like for you guys, 2-liter reactors. So we are increasing the electrode surface for an electrogen more than we needed. So it's never saturated. So we can really test the maximum capacity of our system. And it's working well so far. But I want to tell you about a surprise that we got here. You see here in white that there is something that is not identified. It took us a long time to figure out what the heck was being produced in here. We didn't know if it was a glycerol microbe that was making something new. Only when we grew these bugs together in an electrochemical reactor, something about the electrochemistry was triggering a new fermentation product. And what we found is that looking at the electrode, we noticed that the two microbes were in intimate contact. Rather than having the glycerol microbe doing the fermentation and our electrogen covering the electrode, they were growing very, very close to each other. So a big portion of the glycerol microbe was actually part of this electrochemical process. And when that happened, it shifted its metabolism, and rather than producing ethanol, it started to make something else. Oops, forget about this one for now. It started to make propane diol, 1,3-propane diol. And I won't bother you with metabolism. That can really get all over the head. The important thing is that what we discovered is that this microbe dumps its electrons during fermentation onto ethanol, but also onto the metabolic route for propane diol. These are competitive pathways, and somehow under these conditions it activated this pathway, and we still haven't figured out how that was possible. What we are doing now is simply cutting one or the other and just making sure that the natural process in which it balances the proportion of PDO versus ethanol is no longer a variable for us. So now we're developing an organism that makes only PDO, and the reason why is because PDO is actually a very expensive commodity product. Right now, DuPont is the only company that is making bio-PDO, and is doing it in the normal fermentation, glucose fermentation, but because of the amount of fermentation byproducts that are being produced, it has developed a protected system to separate, very complex system to separate and purify the PDO from all the other ways that is being produced in the fermentation. But what we could have here is a platform in which we only make PDO. So something as simple as a very, very low temperature distillation could be sufficient for us to purify this PDO. So that's one of the projects that we are pursuing because of the money. And this is what I want to end today, to tell you about how we have diversified the projects. The fact that we can make things other than ethanol is something that makes more sense for U.S. biodiesel because of the amount of profit that making a commodity chemical would bring to the industry. So we are focusing our U.S. biodiesel project to making PDO or any other product that now we are trying to reroute now that we understand how this microbe works. So we can add more value to the process, and the biodiesel industry can actually sell at a higher price this byproduct. And we are also reducing the water. That was our mistake. But we decided to start a new project in which we could still think about ethanol for African biodiesel. So one of the projects that we are getting started is funded by the Gates Foundation, and is trying to empower the economy of African countries in which water is so scarce that it will limit really the economy. Think about biofuels, the amount of water. That's why energy is not possible to be manufactured in many, many countries in Africa because of the lack of water. So what we're trying to develop is this type of system, which also will work at smaller scales in African countries. And the reason why is because they have tried to introduce crops for biofuels in Africa, some Western companies. And the effect was devastating. The price of the food that is already such a rare event for African countries, so expensive for them already, just went off the roof. Basically, we were making a very hungry continent even more hungry. That's how. Look at the U.S. grain prices when we started to use them for biofuels. Now imagine the devastating effects in the African continent, for example. But in Africa, in the whole center region of Africa, there are native crops that have been found to be sustainable, that could be cultivated. Agriculture in Africa is in the hands mostly of women, so it is also a very, very social, has a social impact for empowering women in the economy in the African countries. This is just two examples of two small trees that make nuts, and those nuts are very rich in oil, very much like soybeans that we use here. And you can actually just crush them on site and extract the oil to make biodiesel. The problem is that where is the water coming from? You cannot use the water for irrigation and then have enough water to actually use in this type of system at the scale that would be necessary to give them economic independence. So what we are trying to do is follow exactly the same places where these crops could actually have a sustainable environment and economic future to actually adapt our reactors for their biodiesel so we can reduce the water and produce the ethanol that they are going to need for the transesterification reaction. This is the part of the continent where we think we could have an impact. This is also the part of the continent that the Gates Foundation has been focusing on sanitation efforts. I don't know if you've heard about the toilet competition to get the African toilet for sanitation to treat human waste. These are the target areas because of little water availability in these regions. And this is for, by contrast, what would happen if you had something like sorghum for bioethanol. Look how restricted the cultivation would be compared to these native crops. There are also economical studies that show that you can actually have centralized systems. Nothing compared to the big scales that we think about the US. Think about a very small area. Think about Holland as the area in which they could have a sustainable cycle in which a biodiesel plant could be plugged in to the farming activities and to the whole cycle required to sustain this. This study actually includes a biogas plant also to add any amount of energy that can be generated is priceless. But centered to this is water reuse. So that's where we are centered right here, to try to make this center stage more sustainable. So here we have our process. In theory, we could really process just about anything because we can select the substrate and we can screen for organisms that master its metabolism and that feed the fermentation byproducts to our electrogen. So and not only we have the African biodiesel, we have the potential to do just about anything else. And Tom was telling me that he went to the same meeting in Cancun that I went to, and that's where the Gates Foundation awarded projects to fund projects for human waste. So what we are not doing now is actually considering human waste just about any other waste. And what we are doing is trying to have our electrochemical reactors in the latrines that are really the only way, the only toilet that these countries have. We don't have this sewage treatment. We forget sometimes when we use the toilet where that waste goes and how it's treated. But in Africa, we don't have that, and we will never have it for a long time, definitely. So what we're doing is trying to actually couple our electrochemical reactors to the latrines to see if we can actually stimulate the use and generate the biofuel, hopefully. So I'm going to leave it there. I have to thank a lot of people, and I like to present this slide because this was all the projects that I show you. I applied for federal funding. I got recommended for funding with glowing reviews at least twice that I remember, and I never got a dime. And it was the effort state, local effort to support these projects. Until we got the Gates Foundation money, we really hadn't had any external funding. So this is all the foundations and MSU support that we got, state support to get these projects going and take them where we are. So I'm immensely proud and grateful to them for their support, because we would have given up a long time ago, to be honest to you. It started with a Rackham Fund Foundation, just very small grant to get one graduate student designing the proof of concept experiment that we could do it, and it worked. We moved to MSU Foundation and the College of Natural Science that supported my students many semesters because we didn't have money to pay them the stipend. And then this commercialization grant that now is helping us scale up and hopefully bring these platforms to commercialization. So very grateful to all of them. So now I'm happy to take any questions you may have, and I thank you very much for your attention.
Going clear back to the beginning of your presentation, where you talked about the AFEX process and corn stover, have you used any of the product from the AFEX process in your process, or was it just corn stover that you used?
It was the AFEX corn stover.
Okay, so it actually gone through the AFEX process then?
Yes.
Fantastic.
So the reason why is I didn't have time to show you, but I can tell you now. When I first met Bruce and was telling me about the AFEX pretreatment, which is really just a pressure cooker where the ammonium gets inside this very, very tough lignin-containing substrate and explodes it, so the enzymes from the microbes gain access to the cellulose and hemicellulose. That's exactly what other microbes do in nature. So the microbes we're studying cannot attack the lignin that fungi do slowly, but they prepare the substrate in long scale, time scales, exactly the way the AFEX pretreatment does. So I was always very interested in, because of the mimicry of substrate, so there has to be a lot of organisms that are going to love the AFEX corn stover. And sure enough, we did the screening. I actually sent my student to UMass. My PhD lab has a lot of isolates, natural isolates, one of the pioneer labs in making ethanol from all these cellulose degradation processes. And we brought all the organisms in the culture collection and we screened them. We had so many that we could pick and choose whatever was the best. So we went for the one that we could also grow it aerobically to make it easy. We could also, it had the perfect combination of byproducts for our electrogen. I grew more robustly, we knew more about it, and there were so many. And that's because of the AFEX pretreatment. They love it. Because Bruce was telling us, oh, you may have inhibitors. We've seen this with some of the industrial strains. In nature, they're exposed to it, they're prepared for it.
Since we have a number of people who do electrochemistry in the room, would you be willing to say a little bit about the half-cell potential associated with your electrode with the electrogenic microbe?
Yeah, so let's go back to the environment. The type of mineral that these organisms use for respiration has very low potentials in the order of from minus 0.1 to plus 0.1 volts. So minus 100 millivolts to plus 100 millivolts. And anything outside those ranges, it just doesn't do well. So we reproduce those very same potentials in the electrochemical reactor. So what we do is we actually use a potentiostat to poise the potential, and versus I think the hydrogen electrode, I think it's about minus 200 millivolts that we usually use. Very little energy input. That's the thing. But enough to stimulate the activities of this electrogenic microbe to get all the byproducts. So when we think about commercialization, we actually think about just having a small solar panel being used as an energy input, because the voltages that we need are so low that they really do not, I mean, when you see the energy recoveries that I included here, we included the amount of energy input in setting that potential, and it was almost, negligible. Does it answer your question, Paul? Gives you a reference? Tom?
Yeah, I was curious on the biofilm. Have you characterized that? I mean, how thick is it typically? What are the electron transport properties? Things like that?
We have many projects, big project. The NSF project is really focused on that. For example, other groups have done cyclic voltammetry, for example, to single cells, like a monolayer of cells immobilized, like a pectin layer, something like that. And you see the typical cyclic voltammograms, and there is usually, 2 or 3 characteristic peaks that we think are cytochromes of the outer membrane. Because those cells touching the electrodes are going to require very intimate contact, so they usually use metalloproteins, cytochromes, to channel the electrons. That's as much as we know. Genetic studies, you can delete some genes for particular cytochromes, and they're very difficult to interpret because some other cytochrome comes to the rescue. And they may not have the best midpoint potential, but they still, quantity over quality, they still do the job. And it is really difficult to interpret those data. Now think the genome of this bacterium that we're using for electricity production has more than 100 genes encoding cytochromes. The record in all organisms. So many cytochromes, so much iron in those cytochromes that they look red. The cultures are red, just like our hemoglobin, our blood, the same reason. So it's incredible. Now, that's the first layer. As you stack the cells, things change. There is a 10 micrometer distance that electrochemists are puzzled because in those 10 micrometers, you have a signal in cyclic voltammetry that is very different if you try to increase to 20 micrometers. We have a paper that we're about to submit now that uses genetics to show that there is a stratification, that the contribution of the wires and the cytochromes that are in that cement matrix is different because the cytochromes are in a matrix and they have to diffuse. So they're going to be diffuse unlimited. And actually it matches really the theoretical calculations that electrochemists made, is that in those 10 micrometers, that's when diffusion mechanisms, the hopping of electrons from cytochrome to cytochrome is optimal. Anything above that, the cytochromes are there, but they are not reduced. So there is a redox gradient, and there is a pool of cytochromes. If you get too thick, that is useless. And that's where the wires come to the rescue. And the wires the nanoscale are amazing. We talked very briefly about it, but they are like, they have characteristics of carbon nanotubes, but the materials of semiconducting organic materials, for example, is an amazing structure. We have another paper that reveals they are really wires. They transport, just to give you an idea, 10 to the 9th, 10 billion, we're talking billions, electrons per second in a micrometer distance. And it's a protein filament. So it's really a new paradigm. We're going, when we submit papers and we submit grants, but how do they do it? We don't know. We just started. We don't have any counterpart. We don't have a reference for how these wires could connect that. It's a very complex system. So cyclic voltammetry gives you many different.
Well, I'll ask another question while people think. When folks run a traditional ethanol plant, they have to dose it with antibiotics because lactobacillus acts as a predator against the yeast and suppresses. Is there a predator issue with the microbes that form your electrode? In other words, do you have to protect them from other microbes?
Right now we're working with sterility to avoid those issues, but we have considered, as we scaled up, what to do. One of the things that we've been doing, rather than thinking about antibiotics, is to choose organisms that have very low nutritional requirements. So we're using a mineral medium for all our studies. And when you do that, you restrict lactobacillus, for example, from growing. Every now and then we get contamination events, but they're very, very rare because nutritionally you don't support the growth of those organisms. The electrogen is also unique because the moment there is a poised electrode, the right way, it goes there. And it grows and colonizes the surface so fast that it prevents other organisms from growing. So once it attaches, rather than having more cells attaching, it actually grows from the first monolayer all the way up. I'm sure as we scale that, we will have to consider what contamination can do. But so far, so good. I can't tell you much about this because we still have the intellectual property, but we're also trying to choose organisms that have antibacterial effects. They make themselves indigestible because the fermentation ones are the ones that really matter for predation, I think. Not so much. The ones on the electrode, in fact, we're doing runs in which we are trying to reuse the same electrode in several batches, so when we go into commercialization or you think about scaled-up applications, we don't have to clean up the fermenter and set up the electrode again every time. So those are the things that we're considering. We're avoiding antibiotics, by all means.
Other questions for our speaker? Well, let's thank her once again for a very interesting talk.
Thank you.